Can I just get a sip of water before?
Sure.
Good afternoon. I'm Patricia Pinto from the Digital Order. It's a pleasure to initiate this third session in room 3. And I will present now the jury members that are now here with me. We have Nico Cevigli from the Digital Oviedo. We have Luana from PBRC and Gigant. And we have Gauthier and Nico from Digital West Region. And it's a pleasure to say good luck to Alicia that will present. I remember the time, so you have 15 minutes, then we have 15 minutes of the discussion, but we were suggested about 3 minutes per jury member. And if you exceed a bit or you are near to the time, 1 minute, I will do like that, but don't worry. Okay, so it's the floor is yours. Thank you.
Hi everyone, I'm Alicia. I'm very happy to be here. Today, as you can see, my title is very long, but I will basically talk to you about seabirds, a seagull called the aldo ingo, and its distribution beyond its breeding colonies in Catalonia, which is a region in Spain, with insights from citizen science data. Okay, so for those of you who don't know this amazing seabird, it is a Mediterranean migratory species that during the 1970s experienced a sharp decline, and then in 1981 they colonized Punta de la Baña in Delta del Ebro, and the populations start going up, and then with the entrance of terrestrial predators, the populations went down again. It is a highly phylopatric species, and it has subsequent breeding site fidelity. It is currently vulnerable by the IUCN. It is inside the Annex 1 of the European Birds Directive, and Catalonia hosts an important part of the European population. So as I told you before, it is a migratory species, and what this means is that it will nest across the Mediterranean coast, and then will migrate to the west of Africa to spend the winter here, and then they will come back to the coast to breed again. But there is a thing, and it is that this cycle depends on the age of the individual. So the chicks that were born this year will migrate to Africa, and they will stay there until they're ready to breed, and then they will return to the Mediterranean. But the adults will go back and forth every year. And there is an important thing to say here, and it is that not all of the individuals from the population migrate. Then, as with most seabirds, we have a knowledge gap, and it is that most of the data that we know about this species has been gathered during the breeding season and inside colonies. So the inferences that we can make about outside colonies and the non-breeding period are limited. And this is important because understanding the distribution beyond the breeding colonies can help us guide conservation actions. So my objectives were to deepen the understanding of their distribution beyond breeding colonies and throughout the annual cycle using citizen science data and long-term monitoring data, and assessing if distribution differed between breeding and non-breeding seasons, age classes, and individuals from different natal colonies. This is like a bit cut, but well, for the breeding season, I expected more constraints distribution during the breeding period due to breeding constraints and central place foraging. Then, for age classes, I expected juveniles to have broader distributions due to the age-related segregation and exploration refinement hypothesis. If you don't know what these hypotheses mean, don't worry, I will explain them later. And then for natal colonies, I expected that distribution would vary among individuals born in different colonies, with those born in sites with lower resource availability, having broader spatial distributions than those that were born where there are higher quality colonies. So this is my study area, Catalonia, Valencia, and the Balearic Islands. In red, you can see the breeding colonies, and I worked with 2 data sets. One of them was a long-term monitoring mark recapture project from Thesis, who has been bringing birds since 1983. And then I worked with a citizen science database from the Catalan Institute of Ornithology that includes the rings of birds and the sighting date and coordinates. This is the platform where users submit their observations. Here are like the data that you can input. And now we go to one of the most traumatic experiences of my thesis, which was cleaning these huge databases, because I started with 76,000 observations from 1985 to 2026 in 16 different countries. So after going through hell and cleaning, matching records, and quality filtering, I ended up with 12,000 observations from almost 5,000 individuals across 33 years. Now, I classified them in 4 ages, which were chicks, first-year juveniles, second-year juveniles, and adults. We made this classification with the age we had because we knew in what year each individual was born. Then, for the colonies, we found out that our birds came from 42 different colonies, which are the red points. So to facilitate analysis, we grouped them in 11 groups, but we kept 9 due to the sample sizes. Okay, so after all of this work-intensive part, it came the fun part, the spatial analysis. So to be able to analyze coordinates as a 2-dimension response, I calculated centroids per individual per year. And with these centroids, I used them to run kernel density estimates that allow me to see how individuals use space. So I have the 50% contour, which is like the more obscure part, that represents the core areas that individuals use, like the highest intensity use. And then the 95% contour, which are like the outer limits, represent the overall spatial extent occupied by the individuals. Then to see if these areas overlap, I ran 4 overlap indices. And finally, to assess spatial differences, I ran a Permanova, and I also assessed the homogeneity of the spatial dispersion between groups. So, for my results, what did we find during the breeding season? Okay, so we found significant differences between the breeding and the non-breeding season. The 95% distribution was bigger during the non-breeding season, but then something really interesting happened, and it's that when we look at the core areas, they are bigger during the breeding season. And season explained almost 7% of the spatial variation, and this also showed like a lower overlap values across core areas, suggesting that individuals occupy broadly the same regions across the years, but that they differentiate more during, depending on the season. So why is this happening? I am a very drawy person, so here's a diagram. So when seabirds nest, they congregate in a colony and they start laying their eggs. The more they travel away from their colony, the more time they will spend away from their nest, and the higher is the chance that they will lose their offspring to predators. So there's a complication to this, and it is that density is increasing because more and more seabirds are arriving to breed. And then a lot of chicks are born, so adults now forage to cover the needs of their chicks too, so we have an increase in the energetic demands. So competition increases, and this could be explaining the range expansion in core areas that we observe. Now, for the age classes. Age was also significant. It explained the lower proportion of the spatial variation, almost 2%, and we found that younger classes had broader distributions than adults, but also that there was a greater overlap or similarity between individuals among closer developmental stages. And this has This could be explained by ontogeny, which are the changes we go through as we age. And basically, I will talk to you about two hypotheses. The age-related segregation hypothesis that says that there's a spatial shift from juveniles to decrease the competition with adults, and this might have to do with the fact that they are not as good as foragers as adults, and they're not as fit. And then, second, we have the exploration refinement hypothesis that states that there's a greater exploratory behavior during early life stages, and that we, or not we, but like birds specialize as they age. And there's another interesting thing, and it is that second-year juveniles are in preparation for recruitment, can associate with colonies to start learning how to reproduce. So basically, okay, so we will have adults who are super efficient, they have memory and experience in which they rely on, they are specialized but they face breeding constraints, and then we have young individuals who are a bit clueless, they're not as fit, they're still developing their foraging and movement skills, but they don't face any breeding constraints. So, even though age had a low explanatory power, this doesn't mean that it plays a minor ecological role because we're talking about a highly phylopatric species. So this means that age-dependent processes like prospecting, recruitment, and dispersal are linked to colony dynamics. So age might be indirectly contributing to natal colony structure. So now I will talk to you about natal colony structure. So we found significant differences among groups, and it was the one that explained the highest percentage of spatial variation with 16%. And then there's something interesting here. I want you to pay attention to the boards. You see, they're very constrained. Okay. So we found significant differences in distribution among all groups, except for Murcia and Valencia and Columbretes and the Ebro Delta. This was very interesting because there have been previous studies that found that Columbretes was the colony with the lowest quality and the lowest fidelity among individuals, and then the birds from Columbretes were dispersing towards the Ebro Delta. Now, why could this be happening? So as we know, resources are not homogeneously distributed in the environment, so among colonies there are variations in density and resource availability. So if I am a chick, I migrate to Africa, and then I want to come back to my hometown to breed, and I find there's space and there's favorable conditions, I will recruit into this colony and I will display breeding site fidelity, so I will come back after each year. But if I come back and I find my home has been gentrified, then I might need to... Okay. Wait. Oh no, the animation is not there. Well, it was crying. But then, birds need to find another place to nest. So there is a high dispersal. So this species, even though it's very phylopatric, can also disperse if the conditions are not good. And, oh god. So there were ships here, imagine there are ships here. There's also the association with fisheries, which was what I was telling you about when I told you to focus on the boards, because this species is benefiting from trawling and porcine discards. And there are studies that have proved that their distribution is associated with fishing activity. So basically, the effects that we're seeing with natal colony can be an interaction of multiple factors, like the central place foraging, dispersal, the fidelity, and the phylopatry. But perhaps high phylopatry and subsequent breeding site fidelity are the ones that are driving the observed pattern. And also part of the spatial structure that we observed could be related to the seasons and the breeding constraints. So, the limitations of my study. This is a citizen science data, so there were different levels of precision for estimates because there is observer bias and there is also a different observer effort. Chicks also have a short observation window because they migrate, so they're not as comparable to older classes. And we also have enabled sample sizes among natal colonies, which could be due to effort variation, but also to colony productivity and bringing effort across colonies. But this leads us to having lower detection probabilities for individuals that come from colonies that are not in the main monitoring areas. But apart from this data set has many opportunities to use in the future, like improving the survival estimates that we currently have, which are based on inside colony observations, with observations beyond the colony. It could also be a tool for spatial phenology changes detection, especially with the climate change scenario that we're facing right now. And finally, it could help decision makers to identify potential breeding sites and to amplify the conservation measures beyond breeding colonies. So the take-home message would be that season and age influences spatial distribution. Natal colony had the strongest effect, and that understanding spatial ecology beyond the breeding colonies can help us identify key areas for conservation, because migratory species use a variety of habitats throughout the annual cycle, not just during the breeding period where they congregate in nests. And finally, that citizen science, despite its limitations, can complement formal monitoring programs, and it can save us money because it's cheap. Thank you.
Thank you very much, Rachel. Exactly on the 15 minutes, perfect. And now I give my word to the jury. I don't know who wants to start. You can start from here or from there. It can be Nicola. Yeah,
Sure.
I pass.
Well, thank you very much for your presentation. It was really nice. I love the drawings and the sequence of drawings. It was very nice and funny. It helped me to understand what you did. Although I have a question on that actually. What's the time span of the data set?
33 years.
33 years. I didn't get that from the
Yeah, it's
all the way from the 90s.
yes. So the project started in the 80s, breeding, and then it's here. Yes.
Oh, I didn't get. Okay, 33 years. So actually, in your first slide, when you show this diagram of a kind of a time series, you could actually show the data, not just the diagram.
Yeah, I have it. I can show them.
Okay.
I did it also, but
Because it looks to me when you put this kind of figures, it looks like it's a qualitative analysis, no? But if you have the data, you can show it, no?
there
Okay.
is.
Okay. Yeah. So there was an increase and then a decrease. And are you sure it's because of predators, this decrease? Or because there are some species like very similar, like the yellow-legged seagull in the Atlantic, which have decreased a lot in following a very similar pattern to that one.
Well, I think, as always, it must be a combination of several factors, no? One thing that I found very interesting was that they're associating with fisheries. And this is basically, they're getting like a fake signal from the environment because they might be thinking there's enough food for everyone. And maybe the environmental conditions don't fit what the goals are getting from taking the discards. So I think that one of the factors, many factors could be interacting. It could be like a downfall also in like food, interaction with fisheries. They get trapped in fishing lines. We went to the breeding colony and there were many like dead individuals that had been trapped in fishing lines. There's also they're losing the habitat where they used to breed, so they are being pushed away, and they now breed on ports with our suboptimal habitats from them. And there's also like the part of the predators, but yeah, I don't think it's only the predators.
But my question would be, could you actually test this in some way? Do you have the data to test it? Environmental variables, I think you could get it from from satellites or environmental data sets. Fisheries discards, I think there should be some data on that.
Yes. Yes. This would be very interesting in the future too, like to test this, which are the main drivers of the decline of the populations.
Okay. Okay, thank you very much.
Thank you. Alicia, congratulations. Really nice presentation. Perfect to have your enthusiasm and energy, especially the first session after lunchtime. Certainly appreciated it. Beautiful with the drawings. I will make a small question to leave more time for the discussion with my colleagues. But in relation to your last suggestion for citizen science, which I agree with you, it's a very valid approach. But I would like to hear from you if you already had a chance to think. It was not the main goal of your study, but what could be the potential limitations or things to consider when you would recommend citizen science to improve the assessment of these populations?
Okay, so I think that, and this was, I understood this through pain and sweat and spending hours and hours breaking my head trying to clean the databases. There are many errors with citizen science. Like you can commit a lot of errors because it's not only the observer on the field trying to read a ring from the distance. It's also the transcription errors that can happen when the people that are like managing these databases are like transcribing the user's inputs, because sometimes users submit pictures, for example. And then there's someone from the Catalan Institute that is transcribing that. I would say that one of the main things would be to optimize the protocols that we have to obtain citizen science data, to make them the more comparable possible among different projects, especially when we're working with migratory species. Then I would also say like standardizing comments in the databases, because one of the struggles I had with this database is that it was full of different comments, like to say an individual was dead, they used different terminology. It was also in Catalan. I don't speak Catalan. So I had a very hard time translating this in Google Translate or whatever, So like I would say using a common language, making it the methodology like comparable to the ones that were like that can be applied across the places that these species distribute, and then like the standardization, and trying to be as like strong as possible in the data collection process.
Thanks a lot. One thing that there is one that came to mind now, from the observations of the rings and the numbers, I can imagine a lot of errors. Because this happens to me often, that I think I'm reading one number and it's not. Do you think that there could be improved the rings with the use of technology to also avoid that the observations are only done by human sight?
You mean like using AI or something?
I have
like
not.
this?
I sense. I don't know if that would be feasible, but I'm thinking if it would be, or if there is something already like that the rings could have a microchip that is linked to a satellite. Or
I think
like
for
that.
sure, but
I don't have knowledge if this is already.
I think like
exists.
like this could happen. Then we would also have to evaluate the cost of it, because like you see, like there were. I'll show you, like in the ringing database, there were like 60,000 individuals that were ringed. So, but yes, I think this like optimizing the reading process would be nice. But then also, at least for my project, what we had in our favor is that we had the ringing database. So we knew that sometimes there could be an error in the rings. But what we did to take this out of our databases was that we only used those rings that we were 100% certain that were like a perfect match. There's also one thing that I would add, and it's that to ring birds, they use a metal ring and a plastic ring. And sometimes you have the same plastic ring linked to two metal rings, and they're two different individuals. Or the same metal ring linked to two plastic rings, and it's a complete mess. So I would say that one of the things that could be done is like making sure that these kind of things don't happen in the long term.
Well observed. Good. Thank you, Alicia. Congratulations again.
Yeah, thank you very much. And like my colleagues, I really appreciate the way you build up on your artistic skills. And that's an asset for you. Use it. I was wondering about habitat typology that is associated to breeding and non-breeding colonies. So you say that they start breeding in ports, but when you look at your data and the kernel density, it seems that there are some core habitats that are well defined, especially.
Mm-hmm.
Does it correspond to some specific habitats?
Yes, I actually run, it's not included here, I added as an annex in my thesis, but I run like in GIS to see which habitats were preferred, and ports was one of them, and then we also found pastures, which are agricultural areas. So for example, in the Delta del Ebro, there's a big rice plantation, and they go there because there's also the American crayfish, which is an invasive species, and they have been like learning to eat it, and they're now eating this species too. And yeah, like originally they were foraging in the ocean, but now they have also turned to foraging, like using these like human discards, invasive species, and trawling discards. And for the nesting habitat? Were you referring to for the nesting habitat?
Nesting and non-nesting.
Yeah, for the nesting habitat, they used to be nesting in sand dunes or close to the beach, but there's also a huge like anthropic pressure, and these areas have been disappearing. So they are now using like anthropized areas too, like ports.
Okay, and you mentioned on 2 occasions lower quality sites.
Yeah.
Considering what you just said about the fact that they seem to be attracted to highly anthropogenically modified sites, what do you mean by lower quality sites?
That there is a lower disponibility of nesting habitat and also of food. And this relates with what I was saying before about the food subsidies, because ports might be giving them a lot of food, but this is not an honest signal of the state of the ecosystem, because they're basically going to a fast food restaurant and all-you-can-eat buffet, and they're eating a lot. But this has to do with us using trawlers, bringing them like to the surface, and them eating what they can eat. And they actually have like lower nutritional value than their typical or diet. So yeah, this refers to food availability and nesting availability.
Thank you.
Hello, thank you for your talk. Yeah, really clear. I appreciate it again. Yeah, as I say, the use of drawings was really nice. It's a bit of a shame that not everything works, but that's direct for you when that happens. So I had a question about with citizen science data. One of the big problems is that you know where things are, but you don't know where they're not.
Mm-hmm.
And even in time, you can have variation into how many people report and things like that. So I was wondering if you, how do you consider possible variation in terms of basically sampling effort, the amount of people that are working and reporting data in time and in space, basically.
We had this into account from the start. I will show you like we basically selected our study site considering that the Catalan Institute of Ornithology is an institution inside of Catalonia, so most of the users would be based in Catalonia. Because also we had, I'll show you here, these are the whole sightings we had. You can see they're all across, they're all around Spain, but we also have sightings in Africa and even one in New York. So we decided to cut it down to the main areas where the users would be to kind of reduce this sampling effort mismatch. Still, after doing this, there will be differences in sampling effort because users can go to the places, as you said, like they know the birds are, or more accessible sites. And I think that if we didn't account like for a factor of observation effort differences or variation, but I would do this in future like approximations. But yes, it's one of the main things with citizen science data.
Yeah, okay, no, I agree, it's definitely not easy. Another bit related question that I had was about long-term changes. So you have quite a long
I saw that. See.
a long time series. So that's the main, I guess it's kind of an average.
Yeah.
Yes, but I was wondering, did you check if you have, I guess, coupled time and space changes? Because, for instance, you can see some area seems very big when you look at the whole data set, but perhaps it's a population that was once exploited like the south of it, and then moved to the north.
Yeah.
And so it was always in a smaller area. And have you checked that, and how would it affect your conclusion? If are your conclusions robust to that, I guess?
Okay, so like my conclusions are limited by the nature of our data. And they could be, I think they are useful, but they are also, if they are coupled with more like specific studies, like, I don't know, GPS tracking, for example, or knowing if the individuals that are found or cited in Columbrex, for example, or in the Ebro Delta, are they breeding there? Where did they go? Assessing like where is the breeding site fidelity of these birds could help in the future. I did not assess this in this study due to the time limitation, but we did. We were conscious of that, and actually, like the patterns that we're observing in the natal colony could also be due to this of individuals dispersing and then staying in a place. Is this answering your question, or?
Yeah. Yeah, that's one lead. Sorry. Thank you.
Thank you very much. So this is time to present our next speaker. That will be Georgi Urushals. Sorry, I don't know if I'm spelling well.
You need to pronounce E as well. Urushadze.
Thank you.
So,
And we learned about the exponential distribution of polyhedrols, and it's interesting.
Alicia, this is yours. Alicia, this is yours? Yeah.
Yes, I'm sorry.
Okay. So you just put it in like this. Yes, like this.
hello.
Yes, you can hear well.
This is.
Georgi, it will be about polycade and nature-inspired design in graphic 15 minutes. If you are 1 minute from the end, I'll raise my hand. You can go. Thank you.
Hello everybody, my name is Georgi and I'm going to be talking about my master's thesis, which was about understanding the spatial distribution of two building polychaete, Lanicia conchilega, in intertidal habitats to support the nature-inspired designs. So first of all, a little bit of current situation and the background of the study. The coastal ecosystems support over 2/3 of the global population, and they are known to be highly productive and biodiverse, and they provide critical ecosystem services like coastline protection, carbon sequestration, nutrient cycling, and among many others. However, increasing anthropogenic pressures like coastal urbanization and increasing tourism, which is exacerbated by natural pressures like storm intensity, sediment erosion, and sea level rise, have been shown to destroy these environments. And currently, they're the most effective most effective mitigation for this is a hard structure structures, which is like breakwaters and dikes, which have been shown to be effective in a short term. However, they are very costly to maintain and hard to maintain, and they have been shown to disrupt the surrounding environment. Therefore, we need smarter solutions. And NID, which stands for Nature-Inspired Designs, have been shown to be as a solution. So what does NID mean? NID means it's a philosophy of using nature and the environment as a designing element to design a protection or solve a problem. And there's multiple studies that have been showcased using the nature NIDs, different organisms as NIDs, that have been shown to reduce the destructive effects of wave actions, which are mangroves and seagrasses, for example. However, these are very context dependent. So using an organism needs to be based on the environment that we're working with. And my thesis was conducted in Belgian part of the North Sea, where mangroves and seagrasses are not present. Therefore, Lanicia conchilega at 2 building polykit has been shown a promising candidate for this. And it has been shown that it's an ecosystem engineer. It means that it builds biogenic reefs at dense tube aggregations that reduce near bottom current velocity. And they are also biodiversity hotspots, which means that surrounding area are known to increase the biodiversity around them. However, unlike the plant species that I put as examples, this is an animal which has a complex life cycle, which includes free-swimming allophore larvae. Therefore, we cannot just plant them where we want to protect the coastline to use them. Therefore, there remains the gaps to adopt the species as NID. So current gaps include even if we implement them as NIDs, there's no automated monitoring system, and there's no way to track how successful the NIDs are. Also, there's very narrow understanding of where the larvae settles and what are the environmental drivers that drive their settlement. And also, currently, there are no intertidal larval patterns and larval patterns for Lanicia conchilega. Therefore, my thesis was based around 3 objectives to answer each of the gaps respectively. And the first one was to develop a non-intrusive automated monitoring system for the populations of Lanicia to help the success of the NIDs. Second one was to identify the key environmental drivers that govern the settlement of Lanicia larvae and then patch formation. And third one is to identify the seasonal patterns of Lanicia larvae among the intertidal areas. So to talk about the first objective, I needed to design a monitoring methodology that would be non-intrusive to not disrupt the environment and help the NID success. And it needs to be suitable for upscaling because we're protecting a large amount of area, and it needs to process large amounts of data because coastlines are large, so therefore we need to protect a large amounts of area protection. So this project team decided to use drone or UAV, unmanned aerial vehicle photography to track the map, to survey the area. And the drone was used to fly over the Bay of Heist area in Knokke-Heist, Belgium. And the flight altitude was 12 meters. And then the drone took the pictures of 70% overlapping pictures. Therefore, we got this assembled orthomosaic using the software, which gave us the whole area of the surveyed area. Then I needed to design a workflow that would help us automate the monitoring and quantification of the worms. And for this, I applied deep learning model. So I had to design a workflow that includes 3 separate models, deep learning AI models, that will help us give the population estimate and quantify the population. So my workflow was designed the way that it includes 2 branches, branch A and branch B. So branch A is for individually standalone Lanicia worms. So it just delineates individual Lanicias and then gives us count of the worms. And the branch B includes 2 models. So the first model will give us a delineated part of the patch, and the patch includes the worms which are not distinguishable with a naked eye, so we cannot actually count it from the drone imagery. Therefore, we needed to create a separate estimation how much worms are in the patches. And the third model would apply grid points over the delineated patches, and then each grid point would be classified either with a worm title or a background title, and using the literature values, then each patch would be given as their respective density numbers, and the density would be multiplied by the area of the worm patch, which would give us the patch population estimate, and then everything will be summed up, which will give us the combined estimate of whole population of intertidal area. So a little bit of detail how I trained the model. So I cut the in whole orthomosaic in 1 meter squared images to use to train the AI models, and these are the numbers of how much images and how much instance set annotations I did, and some of them were manual, which is for the patch detection. I had to manually delineate patch to train the model what the patch looks like. Then for the first one, it was semi-automatic, so using the pre-loaded model already, it automatically delineates the worms, and then also for the third model, the grid points, where I had to manually annotate which class the point falls on. And these are the results that I got. So in deep learning models, there's 3 metrics that we should look at. So one is precision, which is a metric that takes into account the true positive numbers. The recall that takes into account false negative numbers, and the F1 score, which is the combination of these two. So the F1 score is the harmonic mean, and we should look at this for how accurate the model is performing. And here we see the percentages. And this is one of the examples of what model detected. So this is a patch, and then you see in green the individual worms. And then to visualize my results, I graphed a precision recall curve, and this is also another metric to measure the performance of the model. So it applies a confidence threshold, each confidence threshold, and then it maps mean average precision on each of the confidence thresholds. So for the model 1, I got 87%, and for model 2, 47%. And these are the final outputs for the whole surveyed intertidal area, and in combination, I got more than 76,000 worms present in the surveyed area. So now to move on to second objective, I used the output of my model, which is the number and abundance of the worms in the intertidal area, and then I measured 2 key environmental drivers that have been shown to be governing the settlement of the larvae and the patch formation, which is elevation and sediment composition. And this will help us establish the baseline habitat to implement the NIDs more successfully. So this is the output of the model. This shows us how much worms are in each this much amount of area. And then this is the DEM, digital elevation map, also taken by the drone imagery. And I compared the values of elevation to worm abundance to see the correlation between these two. So first of all, I knew the correlation was not linear, so I applied the Spearman's rule correlation test, and I got the results, and it was significant but weak. But since Spearman's correlation is for monotonic data, then I needed to the weak significance showed me that it's not a monotonic relationship, and there's much more deviance than I expected. So I applied the GAN deviance test, which gave us this graph, and it shows how the elevation is correlated with the worm count, and it only explained the 12% of the worm abundance numbers. Therefore, everything else is explained by other environmental drivers. And then I applied logistic regression, which I treated the worm abundance as categorical numbers, so either worm presence or absence, and that gave us the number that each increase of 1 meter elevation equals the decrease of 30% odds of worm presence. And these are some of the, this is the peak abundance, and this is the average overall in the surveyed area. And now to move on to sediment samples, the project team used the cylindrical sediment cores, and we had 10 different sampling points over the surveyed area, which were randomly assigned. And this gave us the sediment composition, the fractions, and the organic matter data. And I want to show you how the sediment fractions were divided among the samples. And as we can see, all of them are pretty homogeneous. And we can see that all the sediment samples were dominated by the finer sand or mud particles, and the rest of the coarse fractions were really low. And because of this, because of lack of, because of large homogeneity within the samples, we didn't get any significant results. Where this means that we can say that worm abundance is not significantly affected in this small surveyed area. And now let's move on third objective, which is to identify the spatial and temporal larval patterns, which would help us to identify the peak window for NID implementation, and then to help us to identify the spatial variation across the Belgian coast for the best place to implement the NIDs. So there was a campaign from May to October last year, where in these 4 locations alongside the Belgian coast, the project team used the plankton net sampling to grab the planktons, the larvae, free swimming larvae in the intertidal areas, and then these are the results that I got. So I applied two-way ANOVA, and location and month and interaction also showed to be significant. That means that seasonal patterns are very site-specific, so there's no one pattern that each site follows. And one of the key findings include that Bay of Haste area shows the constant highest amount of larval abundance, and also to point out how Bay of Haste and Riverside they have the similar pattern, with Coxside having a reversed peak, as we can see in the later months in autumn. And I want to compare the results for the intertidal area to already published subtidal larval abundance data. So I want you to look at the peaks. So as we can see, the late spring peak is the most common peak for the larval abundance, and then we also see the small second peak in the summer, somewhere in the summer. Usually it's in the late summer, and the autumn peak is almost always present. And now to talk about the key findings. So my study was the first study where I created an automated model for individual level assessment, which we can use to identify the environmental factors like elevation and sediment. And my study was also the first one who identified the larval pattern in the intertidal areas. And how does this help for NID implementation? I identified the area, which was Bay of Haste, and I identified the seasonal pattern, which was the early spring, because late spring has the peak, so we need to implement the NID before that. And how to monitor its success is using my methodology for AI pipeline, and this will help us assess the impact of NIDs for coastal protection. And these are some references. Thank you.
Thank you so much. So exactly 15 minutes, perfect. And now it's time for questions. Maybe we swap the orders. We start with Nico, if you would like.
Yeah.
Yes.
Yeah, so
Of course. So thank you for your presentation. I guess we can start with the sampling for sediments because you didn't found. I mean, it was very homogeneous, and I was a bit surprised by where it was sampled when you look at the map you provided, because it looks like you only. I mean, I don't know what was the constraint, but it's only been checked at the lowest elevation, right? Like they are all in the dark blue.
Well, this was actually randomly selected points, and this was done by not me. So I got this data,
Okay.
and there's also a larger drone flight, whereas this other points fall on. So it's larger area, but I used this because I trained my model on the other drone image, which contains the population, and this is what covers the population. So I didn't extend the whole elevation map to the other points as well.
But were they included in the analysis?
Uh,
The other points that are out of the map.
no, they did not.
Okay.
Because I only worked on this area.
Yeah, I guess probably with a more spread out design, you would have seen more difference in terms of sediment structure, and so perhaps you could have found it.
Yeah. Probably. But this is also a very small area, as we can see. Yeah, here it's only this much, so it's a little small area in the intertidal coast.
Yeah.
Part of the coast. So yeah, it's known to be more homogeneous than. Like we could have expected that it's homogeneous.
Okay. And I guess bouncing back on that, are you confident that the AI you trained to recognize worms and everything, do you know if it would work the same way in like a different place where with different sediments and
For the same.
if you want to deploy it at scale, will you have to retrain another?
You mean for the same organism?
Yeah, same organism.
It will work as long as it's the same organism. It will work as long as like they look like the same organism that the AI model was trained on. So because I also tested, for example, this another part. Here, this is another part where I tested it on different flight in different area.
Okay.
And it has been shown to work. And here I also tested 3 different altitudes of drone flight to see if the altitude matters in protection of the worms.
Okay.
And Giorgio, you're a nerd. With the fonts and the figures,
Yeah.
pixelized figures, I was like, okay, I'm lost in the old video game.
This was the same font for the Python, which
I think
basically
you
was in there. So that's all I could see. That's all I could read or decipher.
Yeah. You need to go back to life, to normal life.
We'll try it after this.
Maybe kind of echoing the last question by Nico. How upscalable is your method? You said you need to be able to upscale it.
Yes.
Is it
Because
upscalable?
Yeah, thank you. So whole purpose of this study was to design the methodology to track the success of NIDs, right? So we need to upscale it. So that was the main reason why we created this. Well, why I created this AI pipeline, and so that I took in mind that it needs to be upscalable. As long as we have big enough processing computer, because this drone orthomosaics are really large in size. So as long as there's CPU that can handle the image, the orthomosaic, then the model will work. So it can work if, in theory, we have big enough CPU for entire Belgian coast. It will work.
Okay.
It will take time, but it will work.
Talking about an idea. I was surprised by the total amount of worms you estimated, 76,000.
That's
Is
why
it a lot?
it is a lot.
To me, it seems ridiculously small.
But this is a really small area of
Yeah, but I mean, did you do the math? Like 76,000 worms on a basically with 40 meter on 40 meters.
Yeah.
I think that's not even
But-
that's sure that's
okay.
that's like 100 worm per square meter or something like that. That's very small, very, and that doesn't seem consistent
Because
with the
there's-
pictures you've showed.
Yeah. But there's, if you look at the whole surveyed area,
Yeah.
transparent parts are where worm was not
Okay,
found.
they're just clustered
Exactly.
in very.
So it's not like the whole area is covered by the worms.
Okay.
As we can see, like almost half of it is just that.
Is it still efficient to reduce wave impact and to?
But that's why we need to implement the NID. We need to. So the project that I was working on, like my master thesis, was part of the broader project, and they're developing the different structures that will help the settlement of the larvae. So there's oh my God. Sorry, I was going. So these are like different structures that they're testing, and I was also part of the campaign where we planted the initial pilot study to detect if this kind of structures help sediment retention and therefore the larval settlement. So there's different. They used mussels, for example. They used artificial Lanicia tubes made from the straws, the stakes, and the bristles, all from the biodegradable material. So yeah, that's the bigger part of the project where they're testing the substrates to help attract more worms. So there's enough worms to help mitigate the impact of.
Thank you, Bill.
Congratulations, dear.
Thank
Fantastic, very interesting presentation.
Thank you.
I want to continue this discussion now. You have the chance to give a thought? It's not the core aspect, the core goal of your research, but a part of the project. So I'm not, don't know, maybe this is being already done in the major project. What should be taken into consideration? What could be the consequences of increasing the worm population for the environment in terms of, for example, interaction with other organisms?
So as I mentioned, Lanichas are biodiversity hotspots. There have been studies done before that have shown that their sediment has much less organic material or organic life around them than patches where the Lanichas are present, because they're known to increase the air supplementation for the deeper layer. So they increase the amount of air in the sediment, which of course that means more life in the area. So that's the whole point of it, because when you put the hard structure, like just the concrete structure to help mitigate the waves, they just disrupt the environment. Basically that makes unlivable that area. And the whole point of NID is to not only protect it, but and to build the structure that will proliferate the life around it. Basically,
That is totally aligned with the discussion and makes total sense.
Yes.
A good alternative to something artificial that will have a huge impact and instead of natural sediment and put hard structures. But what I'm wondering is on the part of other fauna, for instance biofauna, that also lives in the sediment, that interacts with macrofauna such as Lanichi. Do you know if have you had a chance to get further? Do you know if it's being considered? Because it can be positive, it can be negative, but I'm just wondering if this
so
is being thought of. yeah, it succeeds. You succeed, and it starts the implementation. Then you change the environment. You have sediment, as you show. The populations are not with a lot of worms, and the goal is to have much intense density. To have these structures everywhere. This will also change the benthic fauna configuration in another scale.
Yeah, of course, I agree that when we implement them where they are not there, they will be in competition with other neofauna. However, we still need to protect the coastlines, and if this is not a solution, then the solution will be the hard engineering structure, which will be much worse than competing with this. So, I haven't done analysis on how does it affect the meiofauna, but I think just the concrete, like the structures that completely disrupt the environment, I think is much worse than implementing this so that there's some competition. It drives, yeah.
I agree with you. Just important also to
Yeah,
that
definitely.
we also be careful in the considerations. But I will give chance to Nico to also comment on your presentation. But congratulations again, and it's
Thank you so much.
amazing. I saw a picture from Dr. Alves, and I don't know if you're aware how she took it, but she had to use a kite
Yes.
with a camera,
I read that.
and it's fantastic
Yes.
to see the evolution and the amazing.
Yeah, I actually wanted
Yeah.
to talk about how
You did.
the drone idea was born, and it was born by her if I'm saying it correct. Yeah, her study, her PhD. Was it a PhD study? Yeah, I think so.
In 2000, I think, yeah, it was her PhD study.
Yeah, I wanted to say how it inspired this research, but I just didn't have time for that.
No,
But I did cite her. In
no,
the
but I'm
manuscript.
glad that,
Yeah.
that you wanted to talk about it. So that's perfect for our discussion, and we can talk more about it
Perfect.
later in the coffee break. Congrats.
Thank you so much.
Well, congratulations for the work. It's really impressive. I agree that the presentation is a little bit too nerdy for me. Yeah, I appreciate more.
I tried to tone it down. It was much nerdier.
Oh my god! Okay. What is the difference in between your NIDs and the NBS, nature-based solutions?
Nature-based solutions.
Yes. What is the difference? Is there any
So-
difference?
Yes, I think there is, because NID is a physical structure and it's a design. It's a design of the structure. And NBS for me is a nature-based solution, so it's more like an idea of Earth. In my understanding, that's what it's.
Yeah, I've seen the term NBS apply to engineering structures too. So I
Okay.
don't know the difference.
Maybe-
It's a little
Okay.
thing. Don't worry about it.
But I think NID is more like an already pre-existing design, and NBS you apply to the structures to create.
I don't have an answer myself. It's just I'm
Okay.
curious.
This is from the background research. This is what I can say about it.
It's you don't see this kind of studies often when you are able to quantify the population.
Yeah.
The stock
This
of
is
a
the-
given species.
It is the first
intertidal,
one,
So, congratulations for that. It's really nice. About the larval stuff, you measure settlement for how long was that? 1 year or?
It was from May to October, but this research is ongoing, so it's just the window that I took. However, this is the main window because in the winter or colder months, there's for the subtidal population, it has been shown that there's no larval. So we just used the window that was available now, like the data that was available
Do
now.
you know anything about the larval behavior, the planktonic larval duration, all these kind of things? Because those matter, I think,
For the Laniche,
for a niche.
yes. So my thesis was mainly about the
Yeah.
quantification, so I didn't go into details about its life cycle.
No?
However, I did,
I know, but I'm asking this because with such very nice data you have for the adults,
Yeah.
You have some data on the larvae. You can make a stock recruitment model
Mm.
for the whole population, and that would be pretty nice.
Yeah.
Because I think there is a correlation between the seasonal settlement. You have two peaks, right?
There's three, yeah, mainly there's two. There's also like small peak in late summer usually, but it's very small. So yeah, mainly we can say there's two peaks. Like late spring and then autumn peaks.
So if I were you, I would check the seasonality of chlorophyll there.
Ah, yeah, definitely.
And-
I wanted to add that because remember how I said that elevation only explained 12%? So
Yeah.
there's other environmental drivers that can have more effect. And this was just preliminary study to assess some of the environmental drivers, but
You
of course,
might have two peaks in chlorophyll. That's quite common in these waters. So
Yeah.
maybe chlorophyll would be a nice predictor of larval abundance, and then you can couple that with
Yeah.
adult abundance and make a very nice population study.
nice.
Thank you very much.
Thank you for the
Yeah.
suggestion.
So it's now time to pass to our next presenter, Ashanti Rafaela.
I don't know if it's dangerous.
All for
Someone is it's not the best for the students.
small, yes.
Okay. Let's start.
Good afternoon. So again, it's my pleasure to open this session, this fourth session of Room 3. And I will present the jury members that are Nicolas Cegri from University of Oviedo. No, sorry, confusing the Exactly. And Heidi Ancapora from the ATU, and also Katie O'Dwyer from ATU. And also Tiffany, fourth member. So it's my pleasure to call the next speaker, that is Life Borrell Parecera. So remember the timings, you have 15 minutes to talk. I will give you a sign in case you are at the 40 minutes, don't worry. And then the jury members will have 50 minutes, so it's about 3 minutes each. So the floor is yours.
Good luck.
So, good afternoon. My name is Life Borrell Parecera, and today I will present the findings of my master's thesis, which is titled Harbor Porpoise Diet Ecology in the Southern North Sea, Seasonal and Ontogenetic Variation in the Context of Prey Availability, which was supervised by Liz, Sarah, Karen, Jan, and Marlene from ILVO and the Royal Belgian Institute of Natural Sciences. The harbor porpoise is the most common cetacean in the North Sea. It's a small odontocyte with exceptionally elevated metabolic requirements, needing to forage almost continuously in order to survive. Combined with limited blubber reserves, this makes the species particularly sensitive to changes in prey availability. This means that harbor porpoises can integrate signals from the prey community and are therefore widely considered as ecosystem sentinels. Following decades of decline in the southern North Sea, the population began recovering in the late 1990s through a documented southward shift in distribution, which has been linked to changes in prey availability. And porpoises are now present year-round in Belgian waters, which constitute the focal area of this study. The Belgian part of the North Sea is a small but informative system in which to examine harbor porpoise diet, as it is a recolonized habitat at the southern edge of a recovering population, subject to intense anthropogenic activities, and part of a community whose dynamics are relatively well characterized. The main aims of this study was to characterize harbor porpoise diet composition using standard diet indices derived from stomach content analysis, to examine temporal and demographic variation in harbor porpoise diet in the Belgian part of the North Sea across the 25-year dataset, and contextualize observed dietary patterns against known prey availability dynamics in the southern North Sea. It was hypothesized that diet composition would reflect the locally dominant prey community, with seasonal, demographic, and interannual variation broadly consistent with known prey dynamics in the region. For this study, I analyzed a total of 138 harbor porpoise stomachs from animals that were stranded or bycaught in Belgian territory between 2000 and 2025, with most of them having stranded in the coast of Belgium, whose locations are shown on the map. And the necropsies were done by staff from the universities of Liège and Ghent, following a standardized protocol. Prey items were identified from fish, sagittal otoliths, and cephalopod pigs by comparison with published atlases and guides. And prey items were photographed, counted to obtain the minimum number of individuals, measured and paired where possible, and correction factors were applied to the measurements to account for gastric erosion. And finally, prey length and biomass were reconstructed using species-specific regression equations. Prey species were grouped into 9 ecological guilds, and diet composition was assessed, quantified, using 3 standard diet indices, which are percentage frequency of occurrence, percentage by number, and percentage by reconstructed biomass, which were calculated per individual stomach, and then averaged across all stomachs to avoid stomachs with large quantities from skewing the overall diet estimates. Finally, the main statistical framework was divided into 3 parts, which are first Permanova on a Bray-Curtis dissimilarity matrix, of 4th root transformed guild level numerical composition to assess seasonal and demographic trends. Secondly, generalized additive models and with year as a continuous smooth term, and Spearman rank correlations to assess interannual variation, and qualitative comparison against quarterly catch per unit effort indices from bottom troll service in Division 4C of the North Sea to assess consistency with known prey availability dynamics. Seasonal coverage was uneven, with the highest sample being in the 2nd quarter and the smallest being the 4th. Sex, regarding sex, 77 animals were male and 56 were female. And regarding life stage, most of the animals were juveniles, being 104 juveniles and only 33 adults. For cause of death, approximately half of the sample comprised possible or confirmed bycatch, and the overrepresentation of both bycatch and juveniles is due to 2 biases, which is that juveniles are more vulnerable to fisheries interactions, and inexperienced animals are more likely to strand at all. Finally, 104,799 hard prey remains were recovered from the stomach content analysis, which comprised 104,716 fish otoliths and 83 cephalopod pigs. Diet was dominated by 3 main prey guilds, which are gobies, gadoids, and sandils, with secondary contributions from cluebaits and squid. This aligns closely with previous diet studies from the southern North Sea, and supports the characterization of harbor porpoises as opportunistic predators that exploit locally abundant small to medium-sized prey. A total of 28 prey taxa across 21 families were identified, with the most interesting record being the freshwater gudgeon, which was found in a porpoise that stranded in Herentals, which is about 90 kilometers inland from the Belgian coast. And together with the second inland stranded individual, this suggests active foraging in freshwater systems, behavior that has been documented elsewhere in the southern North Sea, but has rarely been reported this far inland. Porter was a strong predictor of dietary composition, revealing a clear sequence of dietary differences in prey availability. In winter and spring, the diet was dominated by gobies, which constitute a year-round prey resource. In summer, there was a peak in sandeel importance, which coincides with their emergence from the sediment in spring and summer, after being largely unavailable from September to March due to overwintering. And then in autumn, there was a large increase in consumption in gadoids, mainly driven by whiting. And this coincides with the period when zero group whiting reaches suitable prey size to become like profitable prey resource. This means that overall gobies provide a continuous baseline diet that is seasonally supplemented by higher energy prey as they become available, and their reduced contribution in summer and autumn is mainly due to higher consumption by other prey guilds, mainly like sandeels or gadoids. At the population level, 3rd quarter diet appears diverse, with multiple prey guilds represented. However, at the individual level, niche breadth was significantly narrower in the 3rd quarter in comparison to other quarters, and 18 out of 28 individuals had only eaten 1 prey guild. And This indicates that summer diversity reflects different individuals targeting different prey rather than broad diets within individuals. And this means that population level generalism is compatible and can arise from individual specialization. This means that a summer population of individual specialists may be more vulnerable to localized prey depletion and may therefore not be able to switch prey. And this result should be tested with a larger sample since it was opportunistic data. I had the samples that I could obtain, but it means that population level diet diversity does not necessarily reflect individual flexibility. Regarding life stage, it explained about 10% of variation in diet, with juveniles highly targeting gobies and adults showing a more even diet composition with greater contributions from sand eels, galoids, and squid. Also, spearmint rank correlations showed an ontogenetic shift in gill diet composition, with goby consumption decreasing with length and sand eel and squid consumption increasing with body size. This is mainly related to differences in foraging capability, as smaller and less experienced animals are targeting easier, demersal, and slower animals that are maybe like gobies, and larger animals targeting faster or more mobile prey that are energetically more rewarding but maybe harder to catch. Although this was not I was not able to test it in the study due to time constraints. The juvenile reliance on low trophic, low energy animals such as gobies may be also consistent with an ecological trap hypothesis, through which inexperienced animals preferentially consume low energy but abundant prey, even though if higher energy prey are available. As for sex, there was no detectable effect on diet, which also when the analysis were restricted to adults, and this may be due to similar prey availability to both sexes or a limited representation of reproductively active females in the dataset. As for during the 25-year study period, there were no significant interannual trends, so dietary composition remained stable and this was seen through the generalized additive models, the Spearman rank correlations, and the Permanova result. And this is remarkable, knowing the known fluctuations in North Sea forage fish, which includes like productivity decline across forage fish stocks and ecosystem regime shift in the early 2000s, and reduced demersal productivity over recent decades. The three different mechanisms may explain this dietary stability, with the first one being the spatial structure in North Sea stock trends, which are mainly driven by its northern component and the southern North Sea, including the Belgian part, remaining relatively stable for key species such as whiting or sand eels. Second, population level buffering through prey switching, even if there is individual seasonal specialization. And third, a decoupling between survey-based abundance indices and prey that is actually accessible to porpoises, as porpoises may target dispersed and small schools of prey that are not well captured by fishery surveys, and productivity can decrease even though if total biomass levels remain above reference. These three mechanisms may jointly explain why no dietary interannual trends were detected, but they should not be interpreted as ecological resilience under future and more severe biomass declines. As for the comparison with diet availability, it was restricted to the first and third quarters due to survey coverage, but for galoids and sand eels, the patterns were mostly concordant, with higher importance in the third quarter and higher catches as well in the third quarter. But for cluepates, the patterns diverged, which is mainly due to limitations in the survey methodology, as juvenile herring and sprat are known to migrate into coastal and estuarine systems during autumn through winter, bottom trawl surveys are also undersampled pelagic aggregations near coastal sites. And harbor porpoises may perform nocturnal pelagic dives to forage on clupates, which daytime fisheries surveys cannot detect. And an independent study based on species distribution modeling corroborated wintertime herring presence in the Battleston part of the North Sea, which is concordant with diet. Overall, this broad comparison indicates that broad agreement with prey composition, with diet composition, but it should not be taken as a strict test of prey selection. Before going into the conclusions, there should be some methodological limitations that should be acknowledged, with first being that stomach content analysis underrepresents prey with fragile otoliths, overrepresents prey with robust ones, and cannot detect entirely soft-bodied prey. Second, the stranding-based sample may not be fully representative of healthy free-ranging animals, as juveniles, animals in poor condition, or animals more vulnerable to fisheries interactions are generally overrepresented, and that the prey availability comparison is constrained by survey seasonal coverage and gear selectivity against small benthic and pelagic species. Future work should include a multi-method framework that combines stomach content analysis with DNA metabarcoding for more taxonomic resolution and to be able to detect soft-bodied prey, with stable isotope or fatty acid profiling to be able to dive into assimilated dietary signals. Also, energy density weighted biomass reconstruction to be able to test the ecological trap hypothesis directly, and biologging or acoustic monitoring of free-ranging animals to resolve foraging behavior beyond what a stranding dataset can show. And for the conclusions, the main conclusions were that harbor porpoise diet was dominated by gobies, gadoids, and sandils, with secondary contributions from clupates and squid. That diet showed clear seasonal and ontogenetic variation, which is consistent with prey availability dynamics and size-dependent foraging constraints. That dietary composition remained stable across the 25-year study period, despite documented fluctuations in North Sea forage fish stocks, and that within individual dietary breadth was narrowest in summer, indicating individual level specialization masked at the population level, and at the level of implication as to how predator and prey relationships are interpreted in a changing environment. Here are the references that I used in this presentation, and thank you for your attention.
Thank you, Lifei. And it was inside the time. It's okay. So now we have the questions from the jury. You can start with the.
Thank you very much. And thank you for this really clear presentation and to respect the timing as well. Okay, I have a few questions. First of all, well, a methodological question, maybe a detail. You mentioned on the diet composition indices that you had made a sort of a correction on the index, the calculation.
Oh,
to avoid it could be a detail. To avoid having a bias linked to bigger stomachs or smaller
like the diet composition
stuff.
indices, yeah.
Do you, how could you do that correction? How does it work?
So I don't remember the exact formula right now,
No, not the exact formula.
but it was just like taking for like in the once the prey species were grouped into the ecological guilds, then for each guild each of the dietary indices were calculated, and I don't exactly remember how, but they were like in the upper part of the the equation. It was like the calculation of the percentage of frequency of occurrence, for example, for each individual stomach, like how many times the prey guild appeared, and then it was divided by. I actually don't really remember right now.
No, I'm not. It wasn't wondering about the formula per se. I was wondering how this could be. I'm not sure how I would correct this. So, but I understand the logic now.
Sorry.
No, that's fine. Then I noticed you mentioned, so you tested the effect of age and sex in the set of samples that you had. So first question, in the case of ages, you noticed that there were differences between juveniles and adults in the composition. So in the future, if you had to select a portion of your samples that would be ideal for your analysis, would you go more for juveniles or more for adult samples? Which one do you think are more representative?
First of all, like samples that are obtained from like a stranding based or bicod, like the stranding network, are already like biased because, and especially with the method that I use, which is stomach content analysis, this method contains a lot of biases. As first, the temporal nature of the method is that since there's like gastric they eliminate the prey remains that they ate after like a few hours, so it only captures the last meal that the porpoise had before dying. So first, like knowing that they are already biased samples that may not be like fully representative of what a healthy porpoise may eat. Maybe like adult samples would be more consistent and more representative of what like a kind of like, I don't know, like normal porpoise eats. And but as they, since I like with the Spearman Guild correlations, it was shown that goby consumption, for example, is not restricted to the limitation to the binomial distinction between juveniles and adults, but rather it's a gradual ontogenetic shift that changes with body size as they are more experienced and are better equipped to be able to capture prey. So I guess like if I mean, maybe if you're checking more like conservation, relevance, how to protect harbor purposes, then maybe assessing like the juveniles is also very important, since they are the ones that will evolve to be adults later on in their lives. So now that I thought about it more, maybe also studying juvenile diet is very important, but also considering with this method that I used that it's really biased.
That's really good.
Thank you very much.
Thank you so much.
Thank you. Thanks very much. Nice presentation and clearly presented. I was wondering, in comparison to previous studies on harbour porpoise, how does the diet compare? Like, is it quite region specific? It seems like their diet is very varied. They have quite a wide diet composition. Can you comment a bit on that?
Yes, so I looked at different studies, mostly focusing on the southern North Sea, so like Belgium part of the North Sea, Netherlands, and maybe also like near the English Channel. And they were mostly reporting a core set of taxa, prey taxa, which are mainly like gobies, sand eels, gadoids, which are whiting and cod also, and clubbaits with herring and sprat. From studies from the 1990s or something like that till studies from 2025, they have all reported like the same set of prey taxa that is dominant in harbour porpoise species. I did see a study that focused more in the German Bight. So they reported more flatfish, for example, and since there is an important nursery area for flatfish there. So I think harbour porpoise mainly reflects like the dominant and locally abundant prey species that are there, at least from like stomach content analysis. So with the temporally discrete nature of it, it's mostly known that since it's the last meal, then it will be mostly close to the coastal area. So with that bias.
Yeah, okay. And then kind of still related to the same topic, you mentioned as one of the limitations limitations that your method only detects prey that have hard parts. But what fully soft-bodied prey items do you expect in the harbour porpoise diet?
Maybe I was.
Like, do you think you're missing something or not in this method?
Maybe it's also more like entirely soft-bodied parts. I was maybe like more referring if maybe they consume like really small squid and it's like mostly like hard parts except
Okay.
for the beak. Then maybe the beak gets lost
Yeah.
somewhere and the rest of the animal is completely digested. So that
Okay.
could be like maybe one example.
Yeah.
Or small fish with like really fragile otoliths that leave no trace whatsoever in the stomach.
Yeah. Okay.
But right now, like no specific prey taxa comes to like
Yeah.
my mind.
Fair. So maybe it's not too much of a limitation then in your method. But
Yeah.
the other thing was on the this diet composition, all the different groups. But do you have any information on their nutritional value? Like, does it matter that they're changing these species in their diet? Or?
So it has been studied, and there are some studies that say that quantity cannot substitute for quality in terms of prey for this harbor porpoise that lives on like the energetic edge of like life. So it has like really high elevated and metabolic demands. So I think as I already mentioned in the future work, in incorporating energy density and knowing like for example clupates, they acquire much more energy from those prey than from gobies, which are like really low energy animals. So it is definitely has been studied that porpoises that feed on like lower energy animals may have to feed more often to be able to sustain their metabolic demands. So maybe how do I say? Since it was not like part of my study, maybe I did not like focus too much on it. But it's definitely like a really interesting question to like further explore and see, for example, like with the ecological trap hypothesis, to see if like juvenile goby heavy diets really, how do you say, have consequences on like juveniles or porpoises that are really relying on those animals to sustain their metabolic expenditure.
Thanks very much.
Evening, thanks for your presentation. Could I just ask you, so I saw that you looked at 138 full stomachs. So that makes me assume that there were more stomachs, but they were empty?
I looked actually at 150 stomachs, and I discarded those since I was focusing on like the diet composition. So since there was nothing in those stomachs, I just took them out from the study, as other papers I had read that did the same thing. But also maybe it would have been also interesting to keep them and see if there was like any seasonal or relationship to like those empty stomachs.
Yeah, that's what I was thinking, because you were also like questioning free dynamics, so it would probably be interesting to, I mean, an empty stomach's your data anyway.
Yeah.
Okay. And then you said that the diet remained stable across 25 years. Do you think that has anything to do with how you like dealt with your data by like averaging all the stomach contents and all of that, and also excluding the empty stomachs?
I mean, definitely the interannual trends part was like a bit more limited, since from the samples I got, there were a lot of differences. Like some years were really well represented, some had just like 4 samples. So it would definitely, they're not like, oh, like diet has remained stable like 100% and it's like all of these for all studies. But it should be acknowledged that I had like a lot of limitations. For example, in 2005, I had like 15 stomachs, but for other years, I had just like maybe 2 or 3. So, and for the intraannual trends part, I actually had to eliminate a lot of some years due to limited sample size. So maybe I over explained and gave too much importance on it, but it's definitely something that should how do you say, that's important to look at. And yeah, sorry.
Okay. And then lastly, could you mentioned individualization in terms of feeding. Could you expand a little bit more on that?
Like individual specialization.
Yeah.
Oh yeah. So do you want to like hear anything specifically, or?
I wanted you to explain to me what you meant there.
Okay,
Expand a
so
little bit more.
as for the population level diet composition, it's in the blue rectangle. You can see that there's like different multiple guilds represented, and they're like quite even. But for the Levin's niche bread, the third quarter was significantly lower than second quarter or fourth quarter, which indicates that most of the individuals were consuming only spray from a single prey guild. So that is important because it may mean that even though like the population is consuming different prey species, individual purposes may consume only one of those. So if in a specific area or region, for example, sand eel abundance suddenly decreases due to an event or like warming or something like that, then those specific purposes that were relying on the sand eel prey may become really affected by the depletion of that prey resource, because they may not actively be able to switch to another type of prey. But as I said, like I think I only had like maybe 20 samples in the summer, so that is also like a big limitation. And as I said, it should be like tested with like more samples to see if this is really a result that is consistent with more purposes, and also with the bias that is from stranding data.
Thank you, Efe. I guess, I have another question about the actually internal patterns. So you tested them statistically with the Permanova as well?
Yeah.
Using year as your explanatory factor?
For the Permanova?
Yes.
I did not include year because I had a very limited sample sizes in some years. So I only conducted a Permanova with year as a sensitivity analysis, but after conducting the general Permanova.
Okay. Yeah, because just from the graph, there seems to be some pretty strong internal variations. Maybe not linear, but you might have some events in the middle of the time series, or so. Yeah, I'm a bit skeptical about the no big internal variation, because depending on how it's done.
For the interannual?
Yeah.
Oh, okay. Yeah, I mean, for the general Permanova, I did not include interannual variation. I just included seasonal, season as with quarter as a proxy, sex, and life stage.
Yeah.
Delay, maybe for delay. Thank you. Thank you so much. Time to call the next speaker, Amelie Winner. We'll talk about the blanket roof improvement, cardiac toxicity. Go ahead. 15 minutes. In case you are at 14, I'll just raise my hand and say, call me.
I hope the microphone works.
Yes.
Enjoy it.
Good afternoon, everyone. My name is Amity Viera, and I'll present my thesis on morphological and structural cardiac plasticity in Atlantic bluefin tuna under aquaculture regimes. Now, before I start, who here has eaten tuna before? Please raise your hand. Okay, that's a good start, but to expand your knowledge on this species, here is some background about Atlantic bluefin tuna, abbreviated ABFT, and its heart morphology. ABFT is a large teleost that swims fast at 45 kilometers per hour and continuously, and they undertake extensive migrations throughout their life. Their cardiovascular system has to be highly efficient and capable of high cardiac output to support their elevated metabolic demands, and the ABFT heart is comprised of 4 chambers, 3 of which are shown here: the sinus venosus prior to the atrium and the ventricle as the primary muscular pumps, as well as the bulbus arteriosus as the outflow tract of the heart. Tuna as active fishes possess a pyramidal ventricle that is shaped like a triangle, and the ventricle consists of 2 myocardial tissue layers, the inner spongy myocardium, which you can see here, and the outer compact myocardium. Tuna as active fishes have a high proportion of compact myocardium in their heart because it has a greater force generating capacity. Due to their high commercial value, there are aquaculture operations for tuna, and in the Mediterranean so far they have been mainly capture-based fattening operations of wild-caught tuna. However, in the past 15 years there's been a transition to closed-cycle aquaculture in land-based rearing tanks where they start rearing larvae. But unfortunately, there should be an exclamation mark here, there is high and unexplained mortality across life stages, which remains a critical bottleneck for closed-cycle tuna farming. Now, in other intensively farmed teleosts, such as salmonids, for example, Atlantic salmon and rainbow trout, cardiac morphological deviations and pathology are causes of mortality. And both of these figures show the more asymmetrical and rounded ventricles with a more misaligned bulbus in farmed compared to wild salmon. Farmed salmon heart and wild salmon heart. And importantly, elevated rearing temperatures is one of the main drivers of cardiac deviations in salmonids. So in this research, we asked the main question, are wild tuna hearts different from farmed tuna hearts? And we hypothesized that intensive aquaculture conditions, which are characterized by elevated temperature, a high level of stress, a different diet, and limited physical activity, impact cardiac morphology and structure and induce deviations in farmed tuna. Second, we focused more on environmental conditions. Temperature can alter cardiac performance, and with ongoing ocean warming and increasingly frequent marine heat waves in the Mediterranean, this might affect wild Atlantic bluefin tuna, and so it is relevant to understand temperature-induced cardiac plasticity. And we hypothesized that elevated water temperatures trigger cardiac remodeling. And lastly, we wanted to know, are cardiac deviations a contributor to the high mortality in farmed tuna? We compared natural and targeted mortalities for this, where natural mortalities were tuna that died spontaneously without any interventions in aquaculture, and targeted fish were randomly selected for sampling and then euthanized. And we hypothesized that cardiac deviations that occur mainly in natural mortalities are a predisposition for this mortality. So we sampled hearts from 3 main groups of tuna. First, farmed juveniles and adults from an aquaculture facility in Spain.
where the tuna are reared in circular tanks with a simulated natural photoperiod and a seawater circulation system, which means that the temperature regimes were similar to wild conditions. Also, the feeding regime was similar to the natural diet of wild Atlantic bluefin tuna across life stages. And now, to give you some insights, here are some videos. From this aquaculture facility, where you can see the dimensions of the rearing tanks that are quite small, with a maximum diameter of 22 meters and a depth of 10 meters. And you can also see that the tuna swim very slowly in there. Then, our second group were adult tuna that were caught in the wild and then reared in fattening cages for a duration of 9 to 11 months. And lastly, we had wild-caught juveniles from 3 locations across Spain and also adults. Furthermore, we had a temperature experiment where farm juveniles were reared at low and relatively elevated rearing temperatures for a total duration of 5 months. There should be a temperature graph here as well that follows the temperature fluctuations across the 5 months. Unfortunately, there has been some complications with PowerPoint, so I'm sorry that you cannot see this. But then we stored the sample hearts in formalin to preserve them and then took photographs in 2 projections, the ventrodorsal and the left lateral projections. And we analyzed these photographs with a standardized morphometric image analysis in Fiji based on an established salmon model with additional parameters that we developed for the heart deviations that were specific to tuna. Now, don't let this confuse you too much, but we quantified a total of 8 shape parameters with different angles and ratios of lengths and areas. And our parameter number 9 was relative ventricular mass, which is ventricle mass divided by the total body mass of the individual fish. For juvenile hearts, we did histological sections to assess the myocardial tissue layers with the software called QPath. Adult hearts were too big for histological sectioning, so we just photographed section cuts of the ventricles and analyzed those with Fiji for our parameter number 10, which was the ratio of compact to spongy myocardium. So what we found is that intensive aquaculture conditions induced early and persistent morphological and structural remodeling in farmed tuna. I'm not showing results plots here per se, but I have them available for questions later. At both life stages, farmed tuna exhibited a more inward curved left oblique ventricular ridge, which is this area of the ventricle that you can see is more curved inward in farmed compared to wild tuna. They also showed a lowered left dorsal ventricular apex, which is this apex, that is lower in farmed compared to wild tuna. Furthermore, farmed tuna had a lower RVM across life stages and less relative compact myocardium area, which is indicated here. And only for the adult stage, we found that farmed tuna have a more misaligned bulbus compared to wild tuna. So overall, these are deviations from the pyramidal shape of the wild tuna hearts toward an overall less symmetrical shape in farmed hearts. And the farmed tuna showed early deviations already at the juvenile stage and then further progressed toward a deviating phenotype at the adult stage. Then, moving on to our temperature experiment, we found that juveniles reared at elevated temperatures exhibited cardiac remodeling. For example, they showed a reduction in relative bulbous size, which you can see in the graph in the middle here. They also showed a more inward curved left oblique ventricular ridge in the high temperature compared to the low temperature group. And this was primarily driven by the targeted individuals, which you can see in light pink here, which were the ones that did not die naturally but survived the experiment. And this suggests that this might be a potentially compensatory response to high temperatures. We also found an interaction for RVM, where at elevated temperatures, again the survivors, the targeted group in light pink, showed an increased RVM, whereas the natural mortalities in light gray showed a decreased RVM at elevated temperatures. Cardiac output generally declines at elevated temperatures, so increasing the ventricular mass might compensate for a loss of function and therefore might be beneficial. So overall, the juvenile tuna heart undergoes shifts in response to thermal conditions, with farm tuna showing morphological plasticity. Now, if we want to pinpoint the drivers of the morphological and the structural deviations that we saw in farm tuna, with this study, we actually can only exclude drivers because thermal regimes did cause cardiac remodeling, as we just saw previously, but they are unlikely to be the primary cause of the deviations between wild and farm tuna. As I told you in the methods part, the temperature conditions and the environmental conditions overall were very similar between wild and farm tuna. Second, stress is something that farmed fish generally experience due to confinement, handling, crowding, and transport. And in salmonids, we know that elevated levels of the stress hormone cortisol lead to specific cardiac remodeling. And we expected to also observe this remodeling in tuna, based on the assumption that stress has a similar effect on tuna hearts. But we did not, so we also excluded stress as one of the primary drivers. Also, feeding regimes did not significantly influence heart morphology and structure, and again, diets were similar for farmed and wild tuna, so that might be why. And that leaves us with the most plausible driver that seems to be limited physical activity and exercise in aquaculture conditions. And the importance of exercise is supported by our observations that generally the fattening cage tuna maintained a heart morphology that resembled the wild tuna. Still, we did observe some gradients from farmed to cage-reared to wild groups for cardiac structural traits, which were RVM on the left-hand side and relative compact area on the right-hand side. And these gradual differences between the groups, so you can see increases from tank-reared to cage-reared to wild groups. We hypothesized that they are potentially caused by a gradient in activity levels, where we assume that wild fish are most active, that the rearing cages restrict activity, and that the rearing tanks restrict activity even more. And with that in mind, our findings indicate that in the fattening cage tuna, the cardiac shape and symmetry has developed normally with their wild background, but that even a short period of activity and limited physical activity that they experience in the cages caused shifts in their cardiac structure toward a more farmed phenotype. So overall, while wild tuna are highly active and undertake extensive migrations, farm tuna are confined in tanks and cages with restricted physical space, high stocking densities, low water current flow velocities, and depth constraints. And these overall restrict their natural behavior, such as high-speed swimming and deep diving, which likely causes the cardiac deviations that we observed. Finally, we could not clearly link mortality in farmed ABFT to specific cardiac traits. This can be due to several factors. For example, some deviations might have a delayed pathology and may represent initially successful compensations. And also the true cause of natural mortality in our samples was unknown and might have been unrelated to any cardiac failure. And furthermore, we found parasites. We found blood flukes of the genus Cardicula, which have been previously documented in ABFT hearts and are a major cause of mortality in farmed southern bluefin tuna. And we found them in both wild, this is a wild heart with parasites, and farmed hearts, where you can see the parasites. And actually in the low temperature group from our temperature experiment, we had 100% of infected individuals with these blood flukes. We did find Cardicula in both natural and targeted mortalities, so a causal link between infection and mortality is unfortunately unclear. But we think that the parasites are a confounding factor as to why we could not clearly identify any cardiac traits that were associated with mortality. So, to conclude, we now know that in farm tuna, aquaculture conditions lead to cardiac deviations. The aquaculture-specific drivers of remodeling remain to be established, but, and there should be another exclamation point here, limited physical activity and exercise likely has a stronger impact than other factors that we looked at in this study. For future studies, we would recommend that a clear link of cardiac structure to function is established, because the actual functional consequences of the deviations that we observed on cardiac performance remain unknown in tuna. And we also don't know yet when, at what stage the cardiac deviations are developed, perhaps already during the larval stage, so that could be investigated. Very importantly, more stressors that are aquaculture specific should be quantified. For example, crowding, exercise and current flow velocity, which we did not quantify in this study. And that can then help develop improved rearing protocols that include forced exercise, for example, with higher current flow velocities in tanks. And maybe also parasite control can be established through treatment options against blood flukes that exist for other tuna species. And altogether, this can be part of a potential strategy to promote healthier cardiac development and to mitigate mortalities in farmed Atlantic bluefin tuna. Thank you for your attention, and I would also like to thank everybody who was involved in this thesis, first and foremost my supervisors, Edurne in Spain and Ida in Norway. Thank you.
Thank you so much. We have your time, 15 minutes exactly. So we will start from the other end of the jury so that you don't run out of questions.
Yeah, thank you for your talk. It was very clear. I'm a bit not sure about the choice. You made the choice for at least one of your results of not presenting the graph, but having like summaries. I tend to like to see the data, especially in like a more scientific context like that.
Mm-hmm.
But maybe not everybody will agree. I had question which is more on the stats because I have no idea about tuna morphology generally.
Mm-hmm.
So how many physical characteristics did you compute in?
We had a total of 10 cardiac traits.
Okay.
That were both morphological, so just from the photographs, and structural from the tissue composition.
Okay. And statistically, you tested an effect of rearing on each of them separately?
Mm-hmm. I can move on to those plots that I talked about earlier, because I have some extra slides for that. So this is an overview for the statistical analysis, where for those 10 cardiac traits, we did comparisons of either two groups, so just comparing farmed versus wild, for example, for the juveniles. And for the adults, where we had the fattening cages, then we compared all three groups. And first we checked the statistical assumptions, abnormality and homogeneity variances, and then we chose the statistical test according to that, with a Bonferroni correction for multiple testing, because we simultaneously tested all of these 10 parameters.
Yeah.
So then we corrected the p-values.
Yeah, that was the main thing I was worried about. But,
yeah, no, I accounted for that.
I don't know. Yeah, that's pretty much it. Yeah, okay. Thank you.
Thank you.
Okay, thanks very much for the presentation. Very, very interesting. So I probably missed it trying to catch up here, like how the fattening cages actually worked. Could you just get back to that, please?
So maybe I can show one of the pictures where we can actually see the cages. Sorry, taking a while.
The suspense is killing me.
I'm so sorry. Okay, there we go. This is the picture that I wanted to show in the middle. So historically, like in the Mediterranean, they culture tuna like this. So they catch adults in the wild, and then they put them into these cages. A lot of these cages are run by private companies, and they just fatten the tuna, so they feed them to increase their market value and to score higher prices on the market. And they keep them in there for a total duration. So the ones in this study were kept there for 9 to 11 months, but it can be from 3 months up to 2 years.
Okay, and then you did not see a change in their heart morphology, is that what I understood?
Yeah, exactly. Okay. So from the photographs, for example, or the morphological traits, where we had those 8 traits that we quantified, We did not see significant differences for a lot of those traits between the fattening cage and the wild groups. But for example, for the cardiac structure, so the tissue composition, for example, we saw these gradients that we thought were interesting. And that, we tried to link with our hypothesis about the limited physical activity and the exercise.
And what was the stocking density like in the pharyngeal? Because when you show the videos, it looks like they don't have a lot of space, but
Yeah.
like, what is the density normally?
I don't know that. That's something that we couldn't quantify. And that we also didn't quantify, for example, for the tanks in the aquaculture facility.
But you imagine that the facility would know how much they have in, no?
Yeah, I just didn't have that data as part of the thesis, or I didn't look at that as part of the thesis, but I think it's something that should very much be quantified.
When you talked about natural mortality, do vets actually look at the fish that have died to try and establish cause of death, or no?
For this study, no.
Okay, so you just basically say natural mortality because they died without you killing them.
Yes, there's some observations from the staff at the facility that I've talked to where they can see, for example, fish jump out of the tanks and they die. A lot of them actually collide with the tank walls, and that is also a big cause of mortality, especially for the smaller tunas, for the juveniles, for example. They also have some malformations in the face, in the eyes, in the jaws that might keep them from feeding. So maybe some of them also die due to starvation, but this has not actually been recorded for all of the samples that I have. So I cannot.
Okay, so if you were to continue this experiment, what would you do next?
I think it would be very interesting to have trials similar to the temperature experiment, except with, for example, 2 different current flow velocities in the tanks. I think that would maybe be easier to implement for juvenile tuna that they keep, since they're smaller, and also the tanks that they keep them in are smaller. But just to have one tank with the current flow velocities that they currently have, and then one tank with an increased current that the tuna have to swim against, and to see if this exercise maybe can change their hearts and promote a more healthier cardiac development. Thank you.
Thanks very much. Nice presentation. Interesting topic. I was wondering, you mentioned, that you don't know yet the links between the heart structure and the function.
Yeah.
But after doing 6 months thesis on this, surely you have some speculations. Where do you think, or what are people proposing as the links between these, and why this focus on heart structure is so important?
So for the thesis, I looked at a lot of the literature that exists for salmon, because salmon hearts have been looked at in detail, and they also did a lot of functional studies. And for example, in salmon, they find that more rounded and asymmetrical hearts indicate a worse swimming performance, or for example, also in trout. They find that it's a worse swimming performance, worse cardiac performance. And so I tried to infer from those findings for tuna, as we also see that the farm tuna have a more asymmetrical heart shape. But for example, some very obvious and specific remodeling, like this more curved inward ridge of the heart, is something that hasn't really been observed for salmonids before. This is novel for tuna hearts. And so it's very hard to say if this is an adaptation, what does this mean for the heart function? So I think to figure that out, some physiological measurements would be really good. For example, to measure your cardiac output, stroke volume, also metabolic rates, for example, resting in maximum, just to see if the fish that have deviating hearts actually perform better or worse.
Okay, but that seems like a kind of a different question to what the industry just wants fat salmon, or fat tuna in this case, or fat salmon in the other case.
Yeah.
So does it matter? Like, my question is more like, who cares about heart morphology if you get a fat salmon at the end, or like a muscular salmon?
Yeah, I think if we link the structure to the function, then perhaps we can also, or it would be possible to figure out more about this high mortality that they have. Because with this study, we didn't really find anything to say, okay, if the hearts have this kind of deviation, that means the tuna is more likely to die. But I think for like economic value of the tuna, it would be good if they didn't have such high mortality. So then,
But is there, like in salmon then, is there links to heart structure and mortality in salmon cages?
yeah, so in salmons there are specific cardiac traits. For example, if the ventricles are more rounded, more asymmetrical, so the general deviations that they find in farmed salmon, they also found that this is connected to mortality, and that the deviations cause mortality.
Okay.
So in salmon that is established much better, and in tuna it's not yet.
Okay, yeah.
Yeah.
No, thanks very much.
Thank you.
Thanks a lot.
Emily, thank you very much. You actually answered a few questions that I was taking notes of during your own presentation, so that's thank you very much. So I'll have just one simple comment now based on one answer you gave. It sounds like you would suggest to open a gym for fish.
Yes.
So in your study you didn't observe an effect of diet, but then if you then changed the current, for example, do you think then the diet should also be different? You tried to copy the diet composition as from the natural diet they would get, but do you think in terms of more the composition, the protein content, for example, or fat content of this diet, do you think that's something that you could play with in the future?
I think so. I have to say I didn't focus very much on the diet in the study, and what they were feeding them with at the facility was of course predetermined and not decided for the study. I think they tried to mimic the natural diet to make sure that the farm tuna did get all of the nutrients that they needed for proper development, etc. And it seems that has worked quite well so far, as there haven't been any differences. But I think if we were to, for example, do trials with forced exercise, etc., then the diet should, I think, be changed according to that, so the fish can still perform well and get all of the nutrients that they need for proper cardiac development, for example.
Okay, thank you.
Yeah.
It would be better to. Oh, okay, so then thank you very much.
Yeah, thank you.
Thank you so much. It's interesting that we are just on time now to follow for the last speaker.
Ja, uitleg aan die presentatie. Zat vol met video's en dingen. Ik denk dat is een ontsluiten. Dat je
Ik ging dus wel in de
Ah, ja.
trekken.
Ja.
Our purpose is Southwest Portugal.
2001.
You can start whenever you're ready.
Okay. Hello, everybody, and welcome to the last presentation for this day. I will talk about spatial and temporal distribution of habitat purposes of southwestern Portugal, insights from a long-term data set. And first, I will talk about why this population, why is it so important? So we have multiple subpopulations of habitat purposes, and I will talk about the Afro-Iberian one, and more specifically about the Iberian coast here. And those are genetically distinct from the others, and Portugal is putting them as critically endangered and in danger of extinction in Spain. And even the IUCN flags them as conservation concerns. So that's not good. Most dangerous thing, bycatch. Unfortunately, very small, shy species, but it gets caught at like unsustainable rates, which is very, very dangerous to them. So to conserve them, that's what's about conservation. We need to know where are they, and there are most likely two ways to get this answer. We have dedicated surveys, which are more done over like a broad structured scale, but they're more on the expensive side. So they often have space and time more coarsely and not very fine scale. But for that, we have opportunistic data or platforms as ferries or fishermen that are taking their routes and submitting them somewhere, or whale watching companies. Those are platforms which are doing like surveys or survey areas very, very often and at very fine spatial and temporal scale at low cost that the other dedicated surveys couldn't meet. And this is also the way that I chose for this study. And I was working at the Algarve coastline where there is like still a big gap of fine scale data. And with this whale watching company I worked together, I developed 4 research questions. First, about their spatial distribution, where do they occur most consistently? Then I was looking at the environmental drivers, which ones are most likely to be associated with them. I went then into the environment versus space, so how much of the variation in harbor purposes is explained by the environmental conditions and by the spatial location. And last but not least, we have the temporal trends. So I will look over 2 decades if there has been a change in occurrence of savage. So, where do we start? Study area. This whole thing was surveyed by the Whale Watch Company, which is a very broad area, and it goes from shelf waters down to very deep waters, about over 1,600 meters. And we have here Cape Saint Vincent, and around here there's a lot outwelling, which means, in other words, it's like a very productive area, very important area. And we have this whale watching company, which I mentioned I worked with. The name is Margarita, which is located here in Sagres. And I got data from 2006 to 2025 from them. Lots of data. 20 years, lots of surveys that I needed to screen. So I needed to apply some filters on it. And after that, I had over 6,700 surveys, over 262,000 kilometers traveled for the surveys, and 260 sightings of harbor purposes. I then took this study area and I applied a 1 × 1 nautical mile grid over it to know, okay, where was the area more surveyed and less surveyed. And I then, those red dots you can see here, are all the sightings. To account for the bias of less and stronger surveyed areas, I developed this encounter rate, so sightings per kilometers, which is a relative index of the occurrence. And with all of that data, I was able to model, map, look at the trends and drivers, and was able to answer the first question. Where do they occur most often? So, we have those 3 areas here around Cape Saint Vincent, around Sagres, and around Salema, where they occur most often. And those areas are present in every season. And they do not distribute to here, 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 and completely diminish. No, they're more shaping in extent how they are. You can see in spring it's a bit smaller, about 71 square kilometers, and a bit more in fall, it's about 120 kilometers. But it's shifting in extent rather than location. And so I was asking the question, okay, it seems structured, what could potentially drive this? So I did a single driver screen first. I was testing the variables in isolation first. 5 of 9 of them were significant. Rock cover, distance to coast, sand cover, coarse cover, and the sea surface temperature. as they were tested in isolation and not accounted for this spatial smooth, so like for the location, they were just as an upper bound. I then modeled them together, so I had a multivariable model where I first put all of them together, all of the variables plus the spatial smooth. I had the so-called like kitchen sink model where I had everything together, but as some of you might know, I cannot model it like that. I needed to account for covariancy and need to take some variables out, and I was taking some out, judged on multiple things, on their model fit, on the single driver screen I did before, on biological interpretability, and ended up with those 4 variables: depth, rock, sea surface temperature, and the spatial smooth, longitude, latitude. And what we can see here is that we have significance in rock really strongly, as well as in the spatial smooth. Sea surface temperature completely dropped. And even here, we can see it's 0.056, so it's not significant, but near significance. But we can see depth was in summer the significant variable. But drawing the attention back to the rock cover, as this is the environmental driver that was overall with under 0.001 p-value, it seems to be very important. Here is the fitted smooth to make it a bit more visual. It's really showing increase. The encounter rate is rising with this hard substrate. What does that mean on the ecological side? It's not that the purpose is saying, rock looks nice, I gotta go there. Maybe, could be tested, but it's most likely that it's connected to their benthnate prey that they're eating that is associated with rock cover. So it's kind of a proxy for prey, often said in other papers. So that is something that definitely should be looked into more. But unfortunately, it's only environmentally uniquely explained 5.5%, location 9.9%, and together 14%, resulting in 29.4%, which means, in other words, we have lots of things that need to be explained in this model. There are lots of things that could explain this model better. So, for example, prey dynamics, they should be a variable for the future that we definitely need to look into, and hydrography. Fine scale hydrography is something that is also found in other marine mammals that could really influence their distribution. Difficult here was that this data, to get it here, is a bit more difficult in the Algarve, and due to time constraints, I couldn't include all of them at once and gather this information, as screening took lots of time for me. But now, decline, encounter rate decline. For lots of biologists, screaming, okay, they're dying, we need to do something. But we have the p-value of 0.004 in the overall model, and we can see it's a bit more steep here in the summer, a bit gentler here in fall, and not significant in spring. In the first association, what I said is often people think, okay, their population is declining. But what if it's not about the animals? What if it is something in the numbers that it's driving that? And I was thinking, okay, we have a company here. Companies usually want to grow bigger, want to extend. So this company did extend over the years, and they went more offshore, which means for this species, which is a species that is more found near shore, that if you go more offshore, this could dilute more or less the found sightings. So I did a consistency cell check, 5 years in the first decade, 5 in the other. And I looked in the end if the significance stayed the same, if I take it counted for this expansion. In other words, no, it did not change anything, the significance was still there, still declined, so there is something in there. But a small little other caveat that I need to mention is I did a relative, as I said in the beginning, I did a relative, not an absolute index here, which means I didn't model the detection. So I cannot talk about the absolute numbers of there or density there, but I can say, okay, they either moved out of this area or they're actually declining. In this model, I cannot say which of those both ways is true, if they're moving out or if they're unfortunately more dying, but that's something. And another caveat mentioned, also on the statistical part, is I was having an offset in my models of 1, which means I was saying for every doubled kilometer, I was saying I will have double the sightings. But it was not at exactly 2 for the doubling, it was 1.8. So close to the 2, which is the exponent of beta is 0.84, which means, in easy words spoken, the decline that we're seeing could be a bit more steep than it really is, but it's not that big of a deal that it's changing everything because it's close to what I modeled with, but better mentioned than thought out through other ways. And the third thing, the course dynamics, sea surface temperature and chlorophyll A. They're all things that are changing. They could even change in minutes, but I needed to summarize them over a month or over season, over years, which would likely understate their role in all of this. But okay, I mentioned now the problems, but I really want to focus on the payoff here because often opportunistic data is overseen, but there's spatial and like fine scale data that they have is something really, really important that we need to look into and we need to work with. What this all means, slowly together, we have this 3 persistent cores that are rather extending, they're not changing their location, they're really shifting in their extent. We likely know that they're connected to the rock coverage, but we must say location was even accounting more for it than the environmentals that I measured. But totally, there's still 70.6% of the variation unexplained. So there's definitely more to discover and more to explain. And the encounter rate declined. It's a relative, not an absolute signal. So that is something we need to know. And that is what I want to say in the end. Those areas, they really should be taken into account for priority areas, for conservation concern, and to know more about them, we need to look into more techniques such as photo identification or hydrophones. Thank you.
Thank you so much. Also well on time, exactly 15. And now maybe we start on this end. Here we go.
Thank you. Thank you very much for your presentation. I have a couple of questions, or more reflection maybe. So you mentioned you had in your data about 260 sightings, right? Over a period of 20 years.
Yes, 20 years, that's correct.
That's a lot of sightings, but especially that we know with these species, it's sometimes you don't really control, or you can't just go out there and find them the day you want to find them. So it is a lot of sighting, and it's a long period of time, which means that you could test all these environmental factors correctly. However, I was thinking, so these sightings mean that per year, on average, you would have maybe 13 sightings. I don't know if you had a look at were there some years or some, the more recent years, there were way more sightings, or the earlier years or something like that. And if you think that's something that could affect your test, your analysis?
Yeah, so I did have a look into that, and this is by month, and this is by year, and we can see that there is not like a real consistent upward trend or downward trend. It really went up and down, and you cannot say, okay, I really surveyed like, oh, here we have Corona, so that's. But we cannot really say, okay, because we surveyed more, we found more, because that's not true here. So I would say in this case that there's no correlation to like found more sightings here or there. But yes, there were only 2 sightings found in winter. Maybe you've noticed winter is completely excluded, as this company doesn't work in the winter time, and it was only 1% of the whole kilometers traveled. So it couldn't give us any statistics here. Yeah.
Okay, that makes sense. Thank you. You mentioned when you were talking about the rock, you mentioned that this could be used in other studies as a proxy of prey.
Yeah.
In this study, do you know? This is pure curiosity. Do you know if you could have access to? Because now that you're seeing a link.
Yeah.
With it, do you know if you could have access to data about potential prey so that you could be a little bit more specific or test a little bit further?
I tried to, but actually there's like a real gap. So the Fae, we need to hear too to figure more out about the diet. There has been one master thesis study. It's not like peer reviewed as well about the data from stranding, so not from living animals there, but there was nothing that I could like work with except for like some strandings where I know what their potential diet in this population is.
That's the difficulty of this kind of study. Very good that you tried that.
Yeah.
Thank you very much.
Yeah, you're welcome.
Thank you very much, Kate.
Thanks very much. That was similar to a question I had on prey and rock cover as well. The other one that I was wondering is you talked about how rock cover was important amongst your environmental drivers, but then when you looked at location was more important. Did you look at location and rock cover at the same time, and would these?
Yeah, that was when I was doing the model together, the multivariable model. And that is like also like here we see the number again of like 29.4%. And
but I mean, or did you look for like a correlation between location and rock cover?
The concurvity. Yeah. There it has been the least. Where is it? No, that was finished. I can go to it directly to show it here. So this is like the concurrency metrics, and we have latitude here and rock here. This was one of them where that was the correlation I found between them. That's like we have some that are overlapping with one, which means like they're really like describing the similar way, and it's 0.61. So there's some correlation, but
yeah, I think that's important. I think that's important to focus and to emphasize when you're explaining the rock cover. You know, because looking at them separately at one stage, but then seeing that they're actually correlated is really important.
Yes, you're completely right. Yeah, that's why I have this also here due to time constraints. So like cutting down everything I didn't know.
Yeah.
But you're welcome. Yeah. You're welcome.
And then I think exactly as you mentioned, I was going to say that like from even already like in your discussion, it might it would have been nice to see already what species from any study on the diet of harbour porpoise anywhere might be more associated with certain habitats, because already like you could just take the results of leaf phase and see where those species tend to are they more occurring in rock habitat or sandy habitat or whatever, you'd actually already be able to find out some of that. I know you mentioned just one master's thesis.
Yeah.
But, there is already information available on harbour porpoise diet, and just you
No,
know,
it
with
is.
speculation. Like, don't be afraid to
Yeah.
step out of your thesis, is what I'm trying to say, and really
Yeah.
look at the broad.
Yeah,
context.
I was looking into that too, and like as I said, there are multiple like subpopulations, like where there's data, especially in the North Sea, it's like very very well studied. But I was afraid to speculate too much on the substrate. Like the substrate is very different in this area than in mine area. So I wanted to rather say, okay, we need to look more into it than saying, okay, we saw this species potentially there in this more sandy area, so this could mean this and that. But I get your point. That's
in the absence of data, we need to
yeah. But that's like my way of thinking how I concluded that yeah.
Thanks very much.
Yeah.
Hi. Thank you. Just a couple of quick questions because I think Nicola has questions on the stats for sure. So yeah, I think very much like Casey said, I was wondering if you connected like your study with other studies in the Iberian Peninsula. You could compare like in terms of distribution and it's similar.
It's so understudied. Like I really had problems of finding anything. What's done is some stranding for like strandings are used for their distribution. At that point, like we are there. Like we're not even that's why I said like the whale watching companies that are spread around the coast is so important because we need to know where the species are. And so it's like a very, very under-sampled, under-surveyed area.
But do you agree that even with the stranding data, I mean, like these are coastal species, like
Yeah,
you
yeah.
mentioned, so probably you know the stranding, like
Yeah.
positioning has some relation to where they are being, you know, distributed anyway.
Yes, there has been, yes, sure, but there is also a bias of fishing, of the bycatch, and especially my supervisor mentioned to me in the middle of Portugal, there is something like they're heavily fishing there. It's like lots of ports are there, so there are like more sightings found there, and it's also mentioned in every of their papers that it's you should be careful with their distribution to stranding, as fishing is like something that's done like for lots of years and like is culturally really important to Portugal. It's a bias, but obviously I mean if they're found there, they must be near the region. I don't think you can go for the more sightings you find. This is the area, the more they're living, because of the bias of the fishing in the area. Unfortunately, like this. But every data you have, you need to use in some kind of way if it's possible and linked to your own. Yeah.
And then lastly, did you take, or I don't think you did because you didn't mention, but who knows, maybe you did, take into consideration observation, like the observer bias, because I mean, if it's a 20-year type thing, you know.
Yes. There is, I assume, like that was part of my thesis I mentioned there too, that opportunistic data has observation bias. Even if you're using different boats, the height where you're standing on the boat. This company actually used mostly the ribs, like the inflatable boats going there, and it's always one or two trained people that are looking without binoculars there, and that is like one side of that kind of observation bias that is part always of opportunistic data that is not done for that purpose. And on the other side, observation data in case of this species is also mentioned, as this species is very shy and small, so there could be lots of animals like far away actually, but you don't see them. This is something why I also mentioned that I didn't do the detection rates, that I cannot say how many we actually missed. So yeah, that's also something important. Yeah.
Yeah, thank you. First of all, I want to give a bit of a congratulation because of, like with my more stats background now that I have this at home, you gave a really good example of how messy variable selection in a model can be, and I think that's a great way to do it because like very often we are like, oh, there is some collinearity, so I just remove the collinear things, and it doesn't work quite like that. So congratulations on that.
Thank you.
And yeah, that's only one. But, yeah. So yeah, talking about all those this variability that is not explained, that you find very often in ecological data anyway.
Yeah.
What I was thinking, what kind of confounding variables could you have on there that are not included in your model? There is one which I was a bit, if I remember well, it's not in there, and I was a bit surprised.
I would be thinking of like what I mentioned as well, like hydrography. That could be something. There could be tides that could be included. The height of the tides, the tides currents, the lunar cycle. I think.
I was thinking something simpler than that, but maybe I missed it.
Yeah, I can show you. I also did depth slope chlorophyll.
Yes. But if you come back to the part where you have like the variance explained and everything with a big circular plot, that one. That one. Then you've got a spatial in there, an environment, which is good. And I was like, where is time?
Where's time?
Yes.
Time as a confound factor for if they're occurring more frequently or not.
Yeah, you definitely have. You show that there is a change in time at the end, and so some of the unexplained variance here could be due to that, right?
Do you mean season or years in time?
Possibly both.
Yeah.
Depending on what you have in the model.
Yeah. This is a good question. I was going more for like environmental explanation itself for their consistent occurrence over the 20 years as it has been stable.
Yeah.
Wait, I maybe I can go back to this. As we don't have winter, and in spring definitely less sampled, it felt like if I would include the season, that there is definitely not like a balanced out possibility, and I wouldn't be confident enough to present this as okay, I can definitely say this is here and there. And same for the years, there's still a difference in the survey effort. So I didn't account for the years, as I said. Like I was more going for why they're there instead of the time being an effect of.
Yeah, no, that makes sense. But I was particularly thinking of possibly because I guess, for instance, like the structure of the bottom, like rock and doesn't move that much. Probably. I don't know if it's totally fixed.
Those are over the 20 years definitely. More.
Yeah. So you could definitely have interaction between those two things. Like at different times, the fixed variable is exploited differently.
Yeah. I will definitely say that's the next thing we probably should
but
then look.
Still you did.
No. More statistics to do. Okay, on it.
And I have another one which is more like on the ecological interpretation side, specifically on the long-term patterns that you see. So if I understood well, but then you mentioned something about relative data, so maybe I'm not sure.
Yeah.
But you see it's pretty stable in spring.
Sorry again?
It's pretty stable in spring, right?
Yeah.
The number of encounters you have. But you've got this decline very pronounced in summer and fall. And I was thinking, look, so, you mentioned that as a decline in population. Potential. And this is where I'm like connecting back to the life cycle of the species. I mean, it's long-lived species.
Yeah.
So the one you see in spring, they're gonna survive until summer and fall and everything.
Yeah.
So I'm not sure about this interpretation then.
I would say something about the spring, and I need to say I would, if I had more time for this thesis, I would have done like some statistical tests about, as I have showed, that spring was not sampled as much as the others. If there could be potentially, as they haven't found any sightings here, if there could be a potential connection between if they actually would have sampled more and if like all of those biases that do come with opportunistic data, if I could account for all of them, and this would go in the hypothetical way, but would make would be logic to me why there's a decline, and there could be as well a decline here, that if they would have sampled more, actually found more of those species as they have been found there as well in spring, that there could be a decline there. So that's why I'm saying there could be the decline of the population, or they moved out. So maybe another thing that could be is that they're going to other feeding grounds or nurturing grounds outside of, because this is like a smaller area. This is not like I cover not whole Portugal. Maybe they're traveling in spring more north, and then they're coming back down. But as I said, this model, I cannot just say which one of the two of them it is.
Nice, thank you.
Is there time for one question?
Yes.
Yes?
Everybody's still not too tired? No problem.
Yes, you're lucky you're that.
I don't know if I missed it, but what is your explanation that you find an effect of depth in summer?
I go back to this.
Yeah.
I was thinking as well the way of prey, sorry the word, of prey that potentially they found in a specific depth more of that specific prey and going there. Or as summer is the peak time of recreational whale watching companies or diving people going out, that they're shifting a bit more far offshore. This could be like another thing. Another thing could be too that bottlenose dolphins and common dolphins especially, there are species found very, very commonly there. And also coming there in summer, that they could be a pressure because there has been a connection found of the dolphins attacking the porpoises, that they're having an effect too on that, which could be also like in the whole thing another explanation of a thing of species crossover, because there's a negative relation found in other waters between the
And is it not possible because you don't find an effect of temperature, surface temperature, that
Yeah.
you just escape the heat, you go deeper?
Yes, that could be definitely as well something that they're doing. They're
Okay.
lots of different ways how that is important. Yeah,
Thank you.
you're welcome.
Thank you so much. And now is congratulations. Thank you for this session at the end of the day. And that's it. And see you tomorrow.
Yeah.
And also thank you for the ones on the streaming online. Thank you.
Okay. Oops.